Hi, I'm Ev.
Hi, I'm Lillian. People with cancer can have their health, time, and quality of life stripped away by this disease.
At the BioMiNT lab, our research focuses on making cancer treatment and remission safer and more accessible to everyone.
The cancer treatment we are working on is chimeric antigen receptor, or CAR T cell therapy, a novel genetic engineering treatment for blood cancers.
CAR T cell therapy treats cancer with a living drug by genetically engineering T cells from patients to target and kill tumor cells, then re-injecting these CAR T cells back into the patient's body. However, the common methods of inserting the CAR transgene into these T cells is usually unsafe and inefficient. Viral vectors can trigger dangerous side effects such as immunogenicity and insertion mutagenesis. Electroporation, a non-viral method, lacks dosage control and has low cell viability. To counteract these limitations, the BioMiNT lab has developed a microfluidic platform called ASOP, or Acoustic-Electric Shear Orbiting Poration. ASOP combines acoustic-driven mechanical shear forces to open pores on the cell membrane, then a gentle electric field to widen these pores. This way, genetic cargo can pass through the cell membrane without viral vectors or using strong electric fields.
The current ASAP device has its limitations, though. The current device is a one serpentine serial channel. This means the vortices to generate the shear forces on the T cell membranes must be filled up one by one before we can apply the electric field. While we wait for the vortices to fill up, the device is continuously pumping, causing some genetic cargo to be wasted.
To fix this problem, we developed a parallel device. In our parallel device, the cells flow through multiple parallel channels at once rather than inching along a single serpentine path. This way, the vortices fill up much faster and less genetic cargo is wasted. We used AutoCAD to design the microfluidic chips and the electrodes beneath the devices. In our preliminary tests, the vortices filled at 1.5 to 2 times the rate of the serial device, and we could expose our cells more uniformly to shear force.
Our parallel ASOP device is also a huge step towards scalability, allowing us to process more cells at once. We are currently using the parallel device to transfect K562 cells, an immortal T cell line, with 500 kilodaltons dextran, so we can test which acoustic and electric field parameters yield the most transfection while keeping the cells healthy.
Beyond the lab bench, increasing the scalability and efficiency of ASOP has real-world applications. The economic barrier to CAR T cell therapy is currently very high. The average price to manufacture CAR T cells per patient is between 300,000 to 500,000 USD. The work we are doing at BioMint aims to make CAR T cell therapy safer and more accessible.
We would like to thank our PI, Dr. Abraham Lee, and our mentors, Manor Mirza and Dr. Kashal Joshi, for their support and mentorship. Thank
Thank
you.
you.